We're talking about hormones made from cholesterol, hormones made from simple amino acids, and huge protein structures, all working to keep, get this, 75 trillion cells in sync.
And that number is the critical context here. 75 trillion cells. It's a number you can't even really visualize properly.
It's astronomical.
It is. And for a multicellular organism like a human to survive, those cells cannot act as rugged individuals. They have to coordinate. They have to act as a unit. They have to adapt to a constantly changing environment. I mean, temperature shifts, food availability, physical stress.
Right.
And while the nervous system is great for immediate point-to-point contact, like touching a hot stove and pulling your hand away before you even think about it, the endocrine system, it handles the long game.
The long game. What do you mean by that?
I mean the long-term adaptation. It regulates metabolism, growth, reproduction, the balance of salts and water in your body. These aren't split-second decisions. They are ongoing, massive projects. And to manage projects that complex, you need a communication system that is equally complex and diverse.
So we have this central theme for today, biochemical diversity. The body uses completely different chemical structures, different shapes, sizes, and properties to achieve very specific physiological goals. Our mission for this deep dive is to figure out how these hormones are built, how they travel through the blood without getting destroyed, which is a hazardous journey, and how on earth they find the right target when they finally get there.
It is a journey from the raw materials, literally the cholesterol and protein in the food you eat, all the way to a signal that can change your DNA expression.
Let's start with the basics then, just so we are all on the same page. What actually is a hormone? I feel like I learned a definition in high school biology that might be a bit outdated. You know, gland A makes chemical B, sends it to organ C, that sort of thing.
You probably learned the classic definition. And to be fair, the definition is still true for the major players like insulin or thyroid hormone. The classic definition says a hormone is a chemical substance synthesized in one distinct organ or gland, dumped into the bloodstream, and transported to a distant target tissue where it exerts an effect.
Right. The thyroid makes thyroid hormone, it jumps on the bloodstream highway, drives to the liver, and does its job. Simple.
Simple, but it turns out to be far too restrictive for modern biochemistry. As we've looked closer, we now understand that the endocrine system is much more local. It's much more fluid. We have concepts like paracrine action.
Paracrine, okay, break that down for me.
This is where a cell releases a hormone, but it doesn't even enter the bloodstream at all. It just diffuses through the fluid between the cells to talk to its immediate neighbor.
So instead of mailing a letter across the country, it's like passing a sticky note to the person at the desk next to you.
Precisely. It's local government. And then there is autocrine action.
Autocrine.
Yeah.
So acting on itself.
Exactly. A cell secretes a hormone that acts on itself. It releases the signal, and then that signal binds to receptors on its own surface to regulate its own activity.
That seems a bit counterintuitive. Why would you shout a command just to hear yourself give it?
It's usually a negative feedback loop. It's a way for the cell to sense its own output. If there is too much signal floating around its own membrane, it's like an alarm bell saying, okay, we've made enough. It knows to slow down production.
A self-regulating system.
Exactly. So the modern definition focuses less on the travel via blood part and more on the specific chemical interaction between a ligand, the hormone, and a receptor. If that specific binding happens and causes a change, it's hormonal signaling.
Okay, so that interaction ligand and receptor leads us perfectly into the first major hurdle this system has to overcome. The sources call it the target cell concept. And honestly, looking at the math in the notes, it is terrifying.
It is a signal-to-noise problem of astronomical proportions, truly.
We have 75 trillion cells. Only a tiny fraction of those produce hormones. And when they do, they release them in concentrations that are almost impossible to visualize. The text mentions the femto to nanomolar range.
Which means nothing to most people.
Right, it's just a number. It's 10 to the minus 9 to 10 to the minus 15 moles per liter.
Those numbers are so abstract, so let's try to ground them. To put 10 to the minus 9, or nanomolar, in perspective, imagine taking a single grain of sugar and dissolving it in an Olympic-sized swimming pool.
Okay.
That is the concentration we are talking about. And for femtomolar, it's like a grain of sugar in 1,000 swimming pools.
That is absurdly dilute. It's basically nothing.
And yet the target cell has to find that single grain of sugar. But it's actually much harder than that because the pool isn't filled with pure distilled water.
Oh, right.
It's filled with soup. The blood is packed with millions of other molecules, structural proteins like albumin, amino acids, sterols, waste products that are floating around in concentrations that are millions, even billions of times higher than the hormone.
So how does it do it? How does a cell know, hey, amidst all this noise, all this chaos, that one tiny molecule floating by is actually a message meant for me?
It comes down to one thing: the receptor. A target cell is defined strictly and only by whether it has the specific receptor for that hormone. No receptor, no effect. The hormone just floats on by, completely invisible to the cell.
It's like having a radio tuned to a specific frequency. If you're tuned to 98.5, you don't hear 101.1, even if that signal is blasting right through your body at the exact same time.
That's a perfect analogy. And these receptors are masterpieces of biochemical engineering. They aren't just sticky spots on the cell surface. They usually have two main functional domains.
Okay.
First, you have the recognition domain. This is the part that physically grabs the hormone, the ligand. It has to be incredibly specific, a perfect lock for the hormone's key.
It has to have a high affinity, right?
A very high affinity, yes. It has to bind the hormone tightly enough to hold on in that sea of other molecules. But, and this is key, not so tightly that it never lets go. The binding has to be reversible.
Because if it never let go, the signal would just stay on forever. You'd never be able to turn it off.
Exactly, and that would be a disaster. Then you have the second part, the coupling domain. Grabbing the hormone is useless if you don't do anything with that information. The coupling domain is what generates the signal inside the cell. It connects the external capture to the internal action.
So I've got the message, and then the coupling domain is what shouts that message to the rest of the cell.
Precisely. It translates the binding event into a biological response.
But here is where it gets really interesting to me. It's not a static system. It's not just a simple question of, do you have the receptor, yes or no? The cell can actually change its mind about how much it wants to listen.
Absolutely. The responsiveness of a target cell is dynamic.
Right.
It's constantly changing based on the environment. One of the key factors is occupancy, how many of the available receptors are currently filled with the hormone. But the cell can also engage in something called downregulation.
Downregulation, what's that?
Imagine you are at a concert and the music gets suddenly painfully loud. What do?
I put in earplugs or just leave the room.
Exactly. You dampen your sensitivity. If a cell is being bombarded by too much hormone for too long, it can actually pull its receptors off the surface, internalize them, and either hide them or destroy them. It desensitizes itself to protect its internal machinery from overstimulation.
Is that what happens in something like type 2 diabetes? I've heard that term used.
That is the classic example. In type 2 diabetes, the body often has plenty of insulin, sometimes huge amounts of it. But the cells, particularly muscle and fat cells, have been shouted at for so long by these high insulin levels that they have downregulated their receptors. They put in the earplugs.
So the signal is there, the insulin is screaming, but the message just isn't getting through anymore.
The message isn't getting through, and I assume upregulation is just the opposite.
Yes. If the signal is very weak, like a whisper in a quiet room, the cell will synthesize more receptors and crowd them onto the surface to try and catch every single stray molecule that passes by. It amplifies its sensitivity.
So the response of the body is this really complex equation. It's the concentration of the hormone plus the number of available receptors plus the internal state of the cell itself plus the intracellular metabolism of the hormone. Sometimes a hormone arrives and the cell has to perform another chemical step to activate it. Or, in other cases, the cell aggressively degrades the hormone the moment it binds, just to stop the signal quickly. It's a constantly shifting balance of send, receive, and destroy.
So we've got the receiver, the receptor. Now we need to look at the messages themselves. The source material divides all hormones into two massive families based on a very simple physical property. Do they like water or do they like fat?
This is the great classification, Group I versus Group II. And this one distinction dictates everything about the hormone: how it is built, how it travels, how long it lives, and most importantly, how it talks to the cell.
Let's start with Group I. These are the lipophilic hormones, the fat levers.
Group I are sort of the VIPs of the endocrine world. This group includes all the steroids like testosterone, cortisol, estrogen, aldosterone, and also the iodothioneins, which are the thyroid hormones, T3/3 and T4/3/04. Calcitriol, which is active vitamin D, and retinoids are in there too.
Why do we call them fat lovers? What makes them lipophilic?
Because chemically, they are mostly derived from cholesterol or have these large non-polar structures that make them hydrophobic. They hate water.
And blood plasma is what, over 90% water?
Right. So the moment they are synthesized, they have a huge problem. They cannot dissolve in the blood. If you just dump them in, they would clump together like oil and vinegar. It would be useless. So they need a boat.
A transport protein.
Yes. They have to hitch a ride on specialized plasma carrier proteins. For example, thyroid hormones ride on something called TBG, or thyroxine-binding globulin. The sex hormones ride on SHBG, sex hormone-binding globulin. Cortisol has its own, called CBG or transcortin.
And this boat does more than just move them from point A to point B, right? It's not just a ferry.
No, it protects them. Because they are wrapped up inside these large proteins, the kidneys can't filter them out of the blood, and the enzymes in the liver can't easily get to them to chop them up. This means group I hormones have a very long half-life. They can circulate for hours or even days.
Okay, so they survive the trip. They arrive at the target cell. Now remember, the cell membrane, the wall around the cell, is a lipid bilayer. It's made of fat.
Which is perfect for group I. Because they love fat, they don't need to knock on the door. They don't need a receptor on the outside. They just dissolve right through the cell membrane. They slip inside like a ghost walking through a wall.
So the receptor isn't on the outside waiting for them.
No. The receptor is waiting for them inside the cell, either floating around in the cytoplasm or, in some cases, sitting right there inside the nucleus waiting on the DNA itself.
And once the hormone binds to that intracellular receptor,
They go straight to the top management. The activated receptor hormone complex moves into the nucleus and binds directly to specific sequences on the DNA. They act as transcription factors. They literally tell the genes, read this section, make more of this specific protein, or stop making that one.
that sounds like a profound fundamental change to the cell's operation.
It is. It's a genomic change. You're altering the very proteins the cell is producing, but because you are printing new machinery, building new proteins from scratch, it takes time. Group I hormones are slow to act. We're talking minutes to hours to days, but the effect is very powerful and long-lasting.
Okay, so now let's contrast that with group 2, the hydrophilic hormones, the water lovers.
These are your proteins, your polypeptides, glycoproteins, and catecholamines like adrenaline. Because they are chemically polar, they love water, they dissolve easily in blood plasma.
So no boat needed for them.
Generally, no. There are some exceptions, but for the most part, they float freely. But because they are swimming around exposed in the blood, they are very vulnerable. The liver and kidneys can grab them and clear them out very, very fast. Their half-life is usually measured in minutes.
Short and sweet. Quick message. And since they love water, I'm guessing they must hate fat.
Correct. So when they get to the target cell, they hit that lipid membrane wall and they bounce right off. They absolutely cannot get inside.
They're stuck on the porch.
Exactly. So their receptors must be on the plasma membrane on the front door. They bind to the outside, and then they have to use a second messenger system to get the word inside the house.
This is where we see all those acronyms that come up in biology like CAMMP, CGMP, or calcium ions.
Yes. The hormone is the first messenger. It rings the doorbell. The receptor on the outside changes shape, and that activates an enzyme on the inside of the wall, like adenylate cyclase, which then starts pumping out the second messenger, like cyclic AMP or CANMP.
And that second messenger is what does the work inside.
That's the little guy that runs around the cell, turning existing enzymes on or off, opening channels, causing immediate changes.
So let me see if I've got this straight. Group I, the fat lovers, they go inside and change the blueprint of the cell by talking to the DNA. Group 2, the water lovers, stay outside and change the machinery that is already running by using a second messenger.
That is a perfect way to put it. Group 2 is all about a rapid response. Think of adrenaline. You get scared, adrenaline hits the receptor, CMP spikes, and within seconds your heart rate goes up and your liver starts pumping out glucose. You don't need to build new heart muscle proteins for that. You just need to stimulate the ones you already have.
It's amazing how the simple chemical nature of the hormone dictates its entire life strategy. Okay, let's get into the nitty-gritty of synthesis. How are these things actually made? The source material takes us deep into the biochemistry of cholesterol derivatives first. This is the steroid story.
And it all begins with cholesterol. We often vilify cholesterol in diet culture, but biologically without it, you would die. Instantly. You would have no cell membranes and you would have no steroid hormones. Cholesterol is the parent molecule for a massive, massive array of critical signals.
But cholesterol is just floating around in the cell membrane or stored in little droplets. How does it get into the hormone factory?
It needs a specialized transport protein called STAR, that's STAR, the steroidogenic acute regulatory protein. Think of STAR as the bouncer at an exclusive club. The club is the inner mitochondrial membrane. STAR is the only thing that allows cholesterol to cross that membrane to get to where the first enzymes are waiting.
And once it's in, that first step is absolutely crucial.
It is the rate-limiting step, the bottleneck for all steroid production. An enzyme in the P450 family, specifically P450cc, or side chain cleavage enzyme, takes that big cholesterol molecule and chops off a piece of its tail. This single reaction converts cholesterol into a new molecule, pregnenolone.
I've heard pregnenolone called the grandmother of all steroids.
That's a very accurate name for it, because once you make pregnenolone, you are committed, you're making a steroid. But which steroid you make depends entirely on which room of the factory you are in. It's like a choose-your-own-adventure book from that point forward.
Let's walk through the adrenal cortex then. This is the outer shell of the adrenal glands that sit on top of your kidneys. The notes say it has different zones, and each zone has a completely different set of enzyme workers.
Right, it's layered like an onion. Let's look at the very outer layer, the zona glomerulosa. The cells here are unique because they possess a specific enzyme called 18-hydroxylase, which is also known as aldosterone synthase. They take that pregnenolone, turn it into progesterone, and then use their special enzymes to manipulate it and create aldosterone.
Aldosterone is the hormone that tells your kidneys to hold on to salt, which regulates blood pressure.
Exactly. But if you move just a few millimeters deeper into the gland, into the middle layer, the zona fasciculata, those cells are different. They do not have 18-hydroxylase. But they have a different worker, 17-alpha-hydroxylase.
So the raw material pregnenolone gets shunted down at a totally different conveyor belt.
A completely different path. It gets hydroxylated, meaning an oxygen and hydrogen group are added at carbon number 17. This path, this specific enzymatic step, is the one that leads to cortisol, the master stress hormone.
It's just fascinating that the raw material is identical. The starting block is the same. It's just about which tools the cell has in its toolbox.
And the sequence of using those tools matters immensely. For cortisol, the sequence of hydroxylation is critical. You have to hydroxylate at carbon 17 first, then carbon 21, then carbon 11. It has to be in that order to produce the final active hormone.
What happens if one of those steps fails? If one of the enzymes is missing?
We see this in a devastating group of conditions called congenital adrenal hyperplasia, or CAH. The most common form is a deficiency in the enzyme 21-hydroxylase. So the assembly line works for the first step, adding the group at C17, but it stops dead at the second step.
So you can't make cortisol, or aldosterone for that matter.
You can't. The body senses the low cortisol and the pituitary gland screams, "Make more." It pumps out ACTH to stimulate the adrenal gland. So the adrenal gland works harder and harder, but the blockage is still there. So all of that backed up raw material has nowhere to go. It spills over into the only pathway left open.
The androgen pathway.
The sex hormone pathway, exactly. So you might have a newborn female infant who lacks cortisol, which is a life-threatening emergency, but has massive amounts of testosterone-like hormones flooding her system, causing profound physical masculinization, all because of one missing enzyme in a complex chain.
Wow. Speaking of testosterone, let's talk about the gonads. Ovaries and testes.
So here, the pathway continues. To make the sex hormones, you need to chop the cholesterol molecule down even further. We started with cholesterol, which has 27 carbons. We went to pregnenolone with 21 carbons. Now an enzyme called 17,020-lase comes in and snips the side chain again, creating the 19-carbon steroids, the androgens.
Like testosterone, DHEA, and androstenedione.
Correct. Now, here's a fact that always surprises people. Biologically speaking, you cannot make estrogen from scratch. You absolutely have to make an androgen first.
Wait, really? So estrogen is just modified testosterone?
Chemically, yes. All estrogens are made from androgens. This happens via a very special enzyme complex called aromatase.
What does aromatase mean in this context? Does it have to do with smell?
No, it's an old chemistry term. In organic chemistry, an aromatic ring is a specific, very stable ring structure with alternating double bonds. The aromatase enzyme takes an androgen, like testosterone, removes a carbon atom, and reconfigures one of the rings into this aromatic structure. This turns testosterone, which is a 19-carbon molecule, into estradiol, an 18-carbon molecule.
So in a way, the female hormone is the final draft, And the male hormone is the penultimate step in that particular synthesis pathway.
In that pathway, yes. And in the ovaries, there is this beautiful cooperation to get this done. It's called the two-cell theory.
How does that work?
The ovarian follicle has two main layers of cells. The outer layer, the theca cells, has all the machinery to take cholesterol and make androgens. It can make androstenedione and testosterone, but it lacks aromatase. It cannot make estrogen.
Okay, so it's stuck with testosterone. It hits a dead end.
But the inner layer, the granulosa cells, has tons and tons of aromatase. It's very good at that last step, but it can't make the androgens from scratch. It lacks the earlier enzymes in the pathway.
So they're completely codependent.
Exactly. The theca cells make the androgens and then hand them over to their neighbors, the granulosa cells. The granulosa cells take the androgens, use their aromatase enzyme, and turn them into estrogens. It's a perfect intracellular relay race. If either cell type fails, the whole system collapses, and you can't make estrogen efficiently.
Teamwork. I love it. Now, there's one more cholesterol derivative that the sources highlight, and it's one that tricked us for a long time. Vitamin D.
Yes. Calcitriol. We call it a vitamin because early researchers discovered that we had to get it from food, like cod liver oil, to prevent the bone disease rickets. But by the modern definition, it is absolutely a hormone. We can synthesize it ourselves, provided we have one key ingredient.
Sunlight.
Sunlight. And the synthesis is described as a relay race across 3 different organs.
Okay, walk me through it. Where does it start?
It starts in the skin. You have a molecule sitting there called 7-dehydrocholesterol, which is made from cholesterol. When UVB light from the sun hits your skin, the energy from the photon actually breaks a chemical bond in that molecule, a process called photolysis. This opens up one of the rings in the structure and turns it into vitamin 3D3 LOX.
And from the skin, it travels to the liver.
The liver acts as the first checkpoint. An enzyme there adds a hydroxyl group at position 25 on the molecule. Now you have 25-hydroxyvitamin 3D33, or calcidiol. This is the major circulating form, and it's what's stored in the body. When your doctor measures your vitamin D levels, this is the molecule they are actually checking.
But it's not fully active yet.
No, it has very low biological activity. For the final, crucial activation step, it must go to the kidney. The kidney possesses the master enzyme of this whole pathway, 1-alpha-hydroxylase. It adds the final hydroxyl group to create 1,025-dihydroxyvitamin 3D33, and that is calcitriol.
And this is the heavy hitter. This is the active hormone.
This is the potent hormone that actually goes to your gut and tells it to actively absorb calcium from your food. And this final step in the kidney is very tightly regulated. The kidney doesn't just make it willy-nilly, it waits for a signal.
What's the signal?
The signal is parathyroid hormone, PTH. If your blood calcium levels drop, your parathyroid glands release PTH. PTH then travels to the kidney and tells the 1-alpha-hydroxylase enzyme to turn on.
So if calcium is low, PTH goes up, tells the kidney, turn on the enzyme, and you make active vitamin D to absorb more calcium.
Exactly. And if calcium is high, PTH levels fall, the enzyme stays off, and you stop making the active hormone. It prevents you from overdosing on calcium just because you spent a sunny day at the beach.
It's an incredibly elegant, fail-safe system. Skin, liver, kidney. All 3 have to be working in concert.
And it really highlights why chronic kidney disease often leads to severe bone problems. If the kidney fails, you lose that final critical enzyme. Doesn't matter how much milk you drink or how much sun you get, you can't activate the hormone you need to absorb the calcium.
Let's shift gears from cholesterol to something much smaller, the tyrosine derivatives. Tyrosine is just a simple amino acid, one of the 20 building blocks of protein found in pretty much any dietary protein, right?
It is, but the body performs some absolutely remarkable chemistry on this one amino acid. We see 2 main, very different classes of hormones come from it. The catecholamines from the adrenal medulla and the thyroid hormones.
Catecholamine pathway, dopamine, norepinephrine, adrenaline, seems pretty linear, like an assembly line.
It is a very clear sequence. You start with tyrosine. The first enzyme, called tyrosine hydroxylase, converts it to a molecule called DOPA. This is the rate-limiting step for the whole chain.
Then DOPA becomes dopamine.
Yes, via an enzyme called dopa decarboxylase. And of course, dopamine is famous as a neurotransmitter in the brain associated with reward and movement. But in the adrenal gland, it's just an intermediate step on the way to something else.
So what's next on the line?
Another enzyme adds a hydroxyl group to dopamine to make norepinephrine, also known as noradrenaline. And finally, and this is a key regulatory point, an enzyme called PNMT adds a methyl group to norepinephrine to create the final product, epinephrine or adrenaline.
The source material mentions a really cool connection here between the stress hormone, cortisol, and adrenaline production.
Yes, this is a beautiful example of intra-organ communication. The enzyme PNMT, the one that makes the final adrenaline product, is induced by high levels of glucocorticoids, by cortisol. Now remember the anatomy of the adrenal gland. Cortisol is made in the adrenal cortex, the outer layer. Epinephrine is made in the medulla, the very center of the gland.
And the blood flows from the outside in.
Exactly. There's a special intra-adrenal portal system that carries blood directly from the cortex into the medulla. So the cells in the medulla are constantly bathed in the highest concentrations of cortisol found anywhere in the entire body.
So when you are chronically stressed, your high cortisol levels are literally fueling the factory that makes adrenaline.
Yes. The long-term stress hormone, cortisol, induces the enzyme that makes the short-term fight-or-flight hormone, adrenaline. It's a way of keeping you primed for danger.
That is incredibly efficient, if a little anxiety-inducing to think about. Now, what about the other tyrosine derivatives? The thyroid hormones, T33 and T44. They're also made from tyrosine, but the process is, well, it's weird. It seems completely unique in all of endocrinology.
It is. For starters, it is the only hormone synthesis pathway in the body that absolutely requires a rare trace element, iodine. The thyroid gland is incredibly efficient at trapping iodide from the blood, concentrating it to levels many times higher than anywhere else.
And the assembly line itself isn't small enzymes, it's a huge protein.
The scaffold for these hormones is a massive protein called thyroglobulin. It has roughly 5,000 amino acids, many of which are chyrosines. The thyroid cells make this huge protein, and then they push it out of the cell into a central storage space called the colloid.
Hold on, so the hormone is being assembled and stored outside the cell?
Yes. It's a very unique strategy. It is stored extracellularly in the center of the thyroid follicle. In that colloid space, an enzyme called thyroperoxidase attaches the trapped iodine to the tyrosine residues on that giant thyroglobulin scaffold. If you attach one iodine atom, you get MIT, monoiodotyrosine. If you attach two, you get DIT, diodotyrosine.
And then they couple up like they're being paired off.
Right. It's like a chemical matchmaking service.
Yeah.
The enzyme grabs two of these already iodinated tyrosines and fuses them together. If you combine a DIT with another DIT, 2 plus 2 iodines, you get T4R, or thyroxine. If you combine an MIT with a DIT, 1 plus 2 iodines, you get T33 triiodothyronine.
But they're still attached to the giant protein scaffold, right?
Yeah.
They're not free yet.
Correct. They are just dangling off the thyroglobulin molecule. This allows the thyroid to store a massive amount of hormone, we're talking weeks or even months worth, in that colloid lake. It's thought to be an evolutionary buffer against periods of iodine deficiency in the diet.
So how do they get released into the bloodstream?
When the thyroid gets the signal to release hormone from the pituitary, the cell has to reach out and drink a droplet of the colloid back up into itself, a process called pinocytosis. It then fuses that droplet with a lysosome, which is basically a cellular bag of acid and digestive enzymes.
The cell's stomach.
Pretty much. The enzymes in the lysosome then chew up the giant protein scaffold, liberating the tiny free 333 and 2333 molecules, which can then diffuse out of the cell and into the blood.
And then they're off to do their work.
Exactly. But here is one last nuance. The thyroid mostly releases T4030s. About 80% of what it secretes is T4 but T43 is the biologically active form. It's much, much stronger. So most of the conversion from the pro-hormone T43 to the active hormone T4, though, actually happens in your peripheral tissues, your liver, your kidneys, where enzymes called deiodonases strip an iodine off T4 to make active T4.
So the thyroid provides the main supply, T4, and the tissues themselves get to decide how much to activate into T42 based on their local needs.
Precisely. It allows for another layer of local control.
Let's move to the biggest class by sheer variety, the protein and peptide hormones.
This brings us right back to the central dogma of biology. RNA makes protein, but for hormones, the protein that comes off the ribosome is rarely, if ever, the finished product.
It's what the text calls the pre-pro hormone.
Correct. It's like flat pack furniture. It's not assembled yet. The pre part is a signal peptide, a short chemical zip code at the beginning of the chain that tells the cell, take this to the endoplasmic reticulum for packaging. Once it gets there, that pre part is cut off. Now you have a pro-hormone.
But even the pro-hormone isn't ready to go.
No. It usually needs to be folded into the correct 3D shape and then often cut again to become active. The classic example that everyone learns is insulin.
Right.
It starts as pre-proinsulin. The signal sequence is removed in the ER, leaving proinsulin. Proinsulin is a single long chain that folds back on itself, and disulfide bridges form to hold that shape. Then it's packaged into vesicles.
And then they snip out the middle piece.
Exactly. Inside the storage vesicle, enzymes come in and cut out the connecting piece, which is known as the C-peptide. This leaves the A chain and the B chain held together only by those disulfide bridges. That two-chain structure is mature, active insulin.
And the text highlighted a really important clinical point about that leftover C-peptide.
Yes, because the final cutting step happens inside the storage granule. When the cell releases insulin, it also releases the C-peptide into the blood in a perfect 1-to-1 ratio, equimolar.
So if I'm a doctor and I want to know how much insulin a diabetic patient's own pancreas is still making,
Yeah.
I shouldn't measure their insulin levels.
Well, measuring insulin can be really tricky if the patient is also injecting insulin as a medication. You can't distinguish the injected insulin from their natural insulin in a standard lab test. But injected insulin is purified. It's just the A and B chains. It has no C-peptide.
So if you measure the C-peptide,
You are measuring only what the patient's own pancreas produced. It's a brilliant biochemical workaround to see endogenous production versus exogenous medication.
that is such a clever application of that basic science. Another example of this cutting process is POMC. This one just blew my mind. Pro-opiomelanocortin.
POMC is the ultimate Swiss Army knife of prohormones. It completely challenges the old idea of one gene, one hormone. Here, a single gene produces one long protein chain. But depending on which tissue it's in and which specific scissor enzymes are present in that tissue, you get completely different and unrelated active hormones.
Okay, walk us through that. What happens in the pituitary?
In the anterior pituitary gland, the enzymes present cut the POMC protein to create ACTH, adrenocorticotropic hormone. This is the hormone that drives your adrenal stress response, telling it to make cortisol.
Okay, so stress.
But contained within the sequence of that same protein is another code. In the intermediate lobe of the pituitary, which is more rudimentary in humans but very active in other animals, ACTH itself can be further cut to make alpha-MSH, or melanocyte-stimulating hormone, which controls skin pigmentation.
So stress and skin color from the same precursor.
Yeah.
And there's more.
Oh, yeah. Further down the POMC chain is a section called beta-lipotropin. And if you cut that piece in a specific way, you get beta-endorphin.
The body's natural painkiller. An endogenous opioid.
Yes. So, from one single precursor protein, coded by one gene, the body can derive a master stress regulator, a pigment regulator, and a powerful painkiller. It implies that evolutionarily, the pathways for stress, pain relief, and even camouflage might be deeply, deeply linked.
It's incredibly efficient genetic coding. Just change the
Absolutely.
scissors. And then we have the renin-angiotensin system. This is another cutting cascade, but this one happens out in the open, in the blood itself, right?
Correct. This is a multi-organ relay system for blood pressure control. It starts in the liver, which makes a large inactive protein called angiotensinogen. It just floats around in the blood waiting.
Waiting for a signal from where?
The signal comes from the kidney. When blood pressure drops or when salt levels are low, the kidney releases an enzyme into the blood called renin. Renin finds angiotensinogen and it snips off a 10 amino acid piece. This creates angiotensin first.
Which is still mostly inactive, from what I understand.
Yeah. Right. It has very little effect. Angiotensin I then flows through the circulation until it hits the lungs. The surface of the blood vessels in the lungs is incredibly rich in another enzyme, ACE, angiotensin converting enzyme. ACE then clips 2 more amino acids off the end of angiotensin I to form the 8 amino acid peptide, angiotensin II.
And angiotensin II is the real heavy hitter.
It is one of the most potent vasoconstrictors the body produces. It squeezes blood vessels to raise pressure almost instantly. It also goes to the adrenal gland and tells it to release aldosterone to save salt and water. It's the master regulator of blood pressure and fluid volume. And of course, ACE inhibitors are some of the most common blood pressure drugs because they block that final activating snip in the lungs.
So we've made the hormones, we've discussed the steroids from cholesterol, the tyrosine derivatives, and these complex peptides. Now they have to get to work. We touched on transport earlier when we talked about group I versus group 2, but I want to dig into this free hormone hypothesis.
This is absolutely critical for understanding both physiology and how we interpret lab tests. As we said, the group I hormones, the lipophilic ones, need those carrier proteins. In reality, these proteins bind up 99% or more of the hormone in the blood.
So only 1% or even less is actually floating free and unbound.
Often much less. For thyroid hormone, T4-1, only about 0.03% is free. And the hypothesis states that only the free unbound hormone is biologically active. It's the only form that can squeeze out of the blood vessel and slip through the cell membrane to find its receptor.
So the 99% that's bound to the protein is just storage. It's just sitting there.
It's a circulating reservoir. It acts as a buffer against sudden changes. If your thyroid were to start working for an hour, you don't immediately become hypothyroid because the carrier proteins will release some of their stock to maintain the equilibrium of the free fraction. It also, as we said, protects the hormone from being cleared by the liver or kidney, which gives it that long half-life.
This brings up the SHBG issue. Sex hormone binding globulin. The sources make a big point that if the levels of the carrier protein change, it can completely mess up the levels of the free active hormone.
Yes. This is a very common point of confusion in medicine. The liver makes SHBG, but the liver's production of SHBG is influenced by other hormones. For example, estrogens and thyroid hormone increase the production of SHBG. Androgens like testosterone and the hormone insulin decrease it.
So let's apply that. If a woman has high estrogen levels, for example, during pregnancy,
She makes more SHBG. There are more seats on the bus, so to speak. This sucks up more of the free testosterone floating around. So her free active testosterone levels can drop significantly, even if her ovaries are making the same amount as before.
and conversely, what about in a condition with high insulin?
In conditions like polycystic ovary syndrome or PCOS, patients often have high insulin levels due to insulin resistance. High insulin tells the liver to start making as much SHBG, so there are fewer seats on the bus.
So the testosterone gets kicked off the bus and is left to float around on its own.
Exactly, you have more free testosterone floating around. This is what leads to the androgenic symptoms we see in PCOS, like acne or hair growth. Even if the total amount of testosterone being produced isn't drastically high, it's a transport and binding issue just as much as it is a production issue.
It's also interconnected. You can't just look at one number on a lab report in isolation.
Never. You have to look at the synthesis, the transport, the receptor status, and the breakdown. It is a completely dynamic, interconnected system.
So to recap this whole journey then, we start with these very simple raw materials, cholesterol or amino acids. We have these specialized factories, the endocrine glands, that use incredibly specific enzymes to craft unique chemical shapes. We release them into the blood, sometimes on special boats, sometimes swimming freely. They have to navigate this sea of 75 trillion cells to find the one specific receptor that fits their shape perfectly. And that one connection triggers a cascade, either changing the cell's DNA blueprint or activating the existing machinery that ultimately keeps us alive and adapted.
It is the ultimate example of biochemical elegance. Biochemical diversity isn't just about having a long list of different chemicals. It's about the system using every chemical property available, solubility, polarity, size, shape, to create a rich and nuanced language. A language that allows your brain to talk to your kidneys and your ovaries to talk to your bones, all through the same shared bloodstream.
It really does make you appreciate what's happening under the hood every second of every day. Which brings us to our final thought.
We've spent this whole deep dive marveling at the incredible specificity of this system, this idea of a key and a lock, a ligand and a receptor. But this specificity, this reliance on shape, is also a potential vulnerability.
How so? What do you mean?
If the entire system relies on the precise three-dimensional shape of a molecule to deliver a critical message, what happens when we introduce synthetic chemicals into our environment that, by sheer coincidence, happen to mimic those shapes?
You're talking about endocrine disruptors.
Exactly. Compounds like bisphenol A or BPA from plastics, phthalates, certain pesticides, and industrial chemicals. Some of these molecules look, from a chemical standpoint, frighteningly similar to estrogen or thyroid hormone.
So they are counterfeit keys trying to get into our cellular locks.
Yes. And if they can fit into the lock, the receptor, even poorly, they can send a false signal, turning on processes that shouldn't be on, or maybe more insidiously, they might just jam the lock, so the real key, the real hormone, can't get in and do its job.
So the very diversity and sensitivity that allows for such complex life might also be the very thing that makes us so sensitive to these new environmental changes.
It is a sobering thought. We are running an incredibly complex biological software on a biochemical operating system that evolved over millions of years in a relatively clean chemical environment. And now we are, in some ways, flooding the hardware with foreign code and expecting it to run without glitches. Understanding the precise mechanisms we discussed today, receptor affinity, synthesis pathways, transport binding, is really the only way we can begin to truly understand the potential risks of those disruptions.
A powerful reminder that our biology doesn't exist in a vacuum. we are permeable.
Always.
Thank you for listening to this deep dive into the endocrine system. Hopefully you have a new appreciation for the wireless network that is keeping you alive and running right now.
It was a pleasure.
We'll see you on the next one.